Hello and welcome again to the microbial world. On this episode, we will be learning about blue-green bacteria, spore-forming bacteria and sheathed bacteria. Kindly subscribe, share, like and send in your comments. Cyanobacteria, commonly called blue-green algae, are true bacteria prokaryotes but share photosynthetic features with plants. They are among the oldest living organisms and fundamentally shaped Earth's atmosphere. They are arguably the most important single group of organisms in Earth's history. Photosynthesis, oxygen production, and global importance. Cyanobacteria are the only prokaryotes capable of oxygenic photosynthesis, meaning they split water molecules and release oxygen. They are referred to as blue-green due to the presence of phycobiliproteins sp, phycocyanin and chlorophyll, giving the characteristic bluish-green coloration. Structures of blue-green bacteria. Outer layers. Mucilaginous sheath, a protective, slimy layer on the outermost part of the cell. Cell wall, a complex, multi-layered wall beneath the sheath, composed of peptidoglycan, polysaccharides, and mucopeptides. Plasma membrane, the inner living membrane of the cell. Internal structure. Cytoplasm, the jelly-like substance filling the cell. Thylakoids, flattened sacs or folds in the cytoplasm where photosynthesis occurs. They are unstacked in cyanobacteria and contain pigments. Pigments include chlorophyll A and accessory pigments like phycocyanin, which gives the blue-green color, phycoerythrin, and carotenoids. Nucleoid, the central region containing the genetic material DNA, which is not enclosed by a membrane. Ribosomes. Smaller ribosomes about 1/3 the size of eukaryotic ribosomes are responsible for protein synthesis and are scattered throughout the cytoplasm. Ecological, environmental and industrial importance. 1. Nitrogen fixation. Crucial for converting atmospheric nitrogen into ammonia. Supports agriculture, for example anabaena in rice paddies. 2. Carbon fixation. Major global contributors to CO reduction. Significant in marine ecosystems. Prochlorococcus equals approximately 20% of world oxygen supply. 3. Harmful algal blooms. HAPs. Some species grow excessively in nutrient-rich water, producing microcystins, anatoxins, saxitoxins. Water discoloration, hypoxia, fish kills. Serious health risks for humans and animals. 4. Biotechnological uses, biofertilizers. Pigments. Phycocyanin for cosmetics and food. Biofuel research. Spore-forming bacteria, primarily Bacillus, aerobic, and Clostridium, anaerobic. These bacteria produce endospores, the most resistant biological structures known. Endospore structure, the ultimate survival weapon. Endospores are non-reproductive, dormant, metabolically inactive. Designed for survival under harsh conditions. Layers of an endospore. 1. Core DNA, ribosomes, enzymes, very low water content. 2. Cortex, peptidoglycan, maintains dehydration. 3. Spore coat, thick protective protein layers. 4. Exosporium, outermost layer. Biochemical features. High levels of calcium dipicolinate KDPA, heat stability. Resistance to UV, desiccation, chemicals, radiation. Clinical importance. Spores withstand boiling, disinfectants, desiccation, alcohol, common antibacterial treatments. This is why hospital sterilization requires autoclaving at 121 degrees Celsius for 15 minutes. To point to sporulation and germination. Sporulation, triggered by nutrient depletion. Harsh environment, process, DNA replication. Asymmetric septation, cortex and coat formation, maturation. Release of the spore, germination, triggered by moisture, nutrients, mild heat. Dormant spore active vegetative cell, 2.3 key genera and examples. Clostridium anaerobic. Clostridium botulinum, botulism most potent toxin known. Clostridium tetani, tetanus. Clostridium perfringens, gas gangrene. Clostridium difficile, pseudomembranous colitis. B. Bacillus aerobic. Bacillus anthracis, anthrax. Bacillus thuringiensis B. key biopesticide. Bacillus subtilis, industrial enzyme production, industrial uses. For the production of enzymes proteases, amylases. Sheathed bacteria form filaments enclosed within a protective outer sheath. Common in aquatic and polluted environments. Structure and function of the sheath. Components. The sheath is composed of polysaccharides, proteins, deposits of iron or manganese oxides. Functions. Protection against predators, toxins, and mechanical stress. Attachment. Strong adhesion to rocks, pipes, and biofilms. Nutrient trapping. Retains organic and inorganic molecules. Structural support. Maintains filament integrity. Genera and their ecological roles. We have Spherotylis, also called the sewage fungus. Forms thick sheathed filaments in nutrient-rich, polluted water. Causes biofouling like clogging of water pipes. Interference in wastewater treatment systems, indicator of organic pollution. B. Leptothrics, which are iron and manganese oxidizers. Sheaths become coated with Fe-cubed and Mn oxides. They are important in iron cycling, formation of iron deposits, influencing water quality, life cycle and movement. Cells inside the sheath are motile when released. Use flagella for movement. Swarm cells. When environmental conditions decline, cells exit the sheath as swarm cells. Swim to a new site, reattach, produce U-sheaths, start a new colony. This ensures survival, dispersal, and colonization of new habitats.